Hi, today I will discuss the topic enzymes. The complete topic will be covered in 4 lectures. In this first lecture, I will be discussing about the properties of enzymes, their nomenclature and classification. So what are enzymes? Enzymes are biological catalysts. They increase the rate of biochemical reactions. And enzymes are found in all the living cells in animals, plants and microbes. Without enzymes, life is not possible. About 1/4 of the genes in human genome encode enzymes. And if there is a mutation in the gene, it may result in deficiency, total absence or excessive activity of an enzyme leading to inheritable genetic disorder. Enzymes are also used as diagnostic tools to diagnose diseases. For example, if in a blood sample the level of cardiac enzymes is elevated, it means that there is myocardial infarction or damage to heart muscles. Or if there is increased level of liver enzymes in the blood sample, it may indicate inflammation in the liver or a disease in liver. Many drugs interact with enzymes to treat metabolic disorders. Enzymes can be produced artificially through microbial fermentation and are used in chemical industry, food processing industry and in agriculture. Now we come to the properties of enzymes. I told you that enzymes are biological catalysts. They increase the rate of biochemical reactions. All the enzymes are made up of protein except few ribozymes which are catalytic RNA. Many enzymes require a non-protein coenzyme or cofactor for their catalytic activity. Enzymes like proteins have high molecular weight ranging from 12,000 to more than 1 million. Enzymes are very specific for their substrates. The substrates bind at the active site in the enzyme molecule. Enzymes increase the rate of chemical reaction without the formation of byproducts, and they function in dilute aqueous solutions under very mild conditions of temperature and pH. If we increase the temperature or change the pH, or under non-physiological concentrations of salt, organic solvents, urea or other chemical agents, the enzymes get denatured. Denaturation of enzyme means that they lose their catalytic activity. And why they lose their catalytic activity? Because enzymes and proteins have a proper folding of amino acid chain into secondary structure and tertiary structure. Oligomeric proteins have a quaternary structure, and under denaturing conditions, the enzymes lose their conformation or the native protein structure, thereby losing their catalytic activity. Enzymes catalyze reactions in both the directions. They catalyze forward reaction and also reverse or backward reaction. During enzymatic catalysis, the substrate molecule binds at the active site for a brief period of time, transiently, and after catalysis, the product is released. Enzymes increase the rate of reaction, but they do not alter the equilibrium. Now we come to the nomenclature and classification of enzymes. International Union of Biochemistry and Molecular Biology has classified the known enzymes into 7 classes on the basis of reactions they catalyze, substrates transformed, and the products formed by the enzyme. Earlier there were 6 major classes. In 2018, 7th class translocase was added. Currently there are 6,476 enzymes known, and all these enzymes are identified by 4-digit enzyme commission number known as EC number. These are the 7 classes of enzymes: oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases. There are 1908 enzymes in the class oxidoreductase, and these catalyze oxidation reduction reactions by transfer of electrons, hydrogen or oxygen. Second class is transferase with 1929 enzymes. Transferases catalyze group transfer reactions. Hydrolases have 1314 enzymes. These catalyze hydrolysis reaction, transferring the functional group to water. Digestive enzymes belong to the class hydrolases. The fourth class is lyases with 708 enzymes. Lyases catalyze the addition of groups to double bonds or formation of double bonds by removal of groups. The fifth class is isomerases with 304 enzymes. Isomerase transfer groups or atoms within the same molecule. So these are known as intramolecular transferases, thereby converting one isomeric form into another. The sixth class is ligases with 223 enzymes currently. Ligases catalyze the formation of carbon-carbon, carbon-sulfur, carbon-oxygen and carbon-nitrogen bonds by condensation reactions coupled to ATP cleavage, that is release of energy from ATP. The seventh class is translocase with 90 enzymes. These catalyze the movement of ions or molecules across membranes or their separation within membranes. Now we come to the examples of these classes. First class is oxidoreductase with the example lactate dehydrogenase. The coenzyme used in this reaction is nicotinamide adenine dinucleotide or NAD. NADH plus H plus is oxidized to NAD plus and pyruvate is reduced to lactate. The example of transferase is hexokinase. Hexokinase transfers phosphate group to glucose, forming glucose 6-phosphate. Third class is hydrolases. Sucrase belongs to hydrolases. It hydrolyzes sucrose into glucose and fructose using water. Next class is lyases. Example is histidine ammonia lyase. It breaks the carbon-nitrogen bond, creating a double bond. Isomerases, example is alanine racemase. It converts L-alanine into D-alanine by intramolecular transfer of atoms. The example of ligase or synthetase is glutamine synthetase. Glutamate and ammonia combine to form glutamine and the energy is used from ATP. Translocases transfer molecules or atoms across membranes and the example is adenine nucleotide translocase, which is involved in the transfer of ATP and ADP. All the enzymes are given a 4-digit EC number or enzyme commission number. Each enzyme has a common or trivial name. For example, hexokinase is the common name. Its formal systematic name is ATP glucose phosphotransferase, which indicates that hexokinase catalyzes the transfer of phosphoryl group from ATP to glucose. The EC number of hexokinase is 2.7.1.1. The first digit 2 denotes the class name transferase. The second digit 7 denotes the subclass phosphotransferase. The third digit 1 denotes that it is a phosphotransferase with alcohol or hydroxyl group as an acceptor, and the last digit 1 denotes that D-glucose is the phosphoryl group acceptor. Nomenclature Committee of the International Union of Biochemistry and Molecular Biology maintains a complete list and description of thousands of known enzymes. You can visit this site to know more about the enzyme nomenclature. Thank you so much.